Welcome to the PubMed Daily Review for May 22nd, 2026. Today we scanned 3,817 new open access papers. Here are the top 7 selected for detailed analysis. Trend 1 of 10. Clinical Surgical Orthopedic Research. This trend investigates clinical outcomes, patient factors, and surgical management strategies in orthopedic procedures. This cluster covers 367 papers. Representative work includes clinical and demographic factors associated with baseline PROMIS categorization in shoulder and elbow patients, surgical management of Charcot neuroarthropathy involving the hindfoot and midfoot, a case series, and biomechanical evaluation of structural integrity after tibial plateau, leveling osteotomy in dogs using finite element analysis. Trend 2 of 10. Cellular and molecular biology mechanisms. Unraveling cellular and molecular mechanisms that govern diverse biological processes, development, and disease. This cluster covers 441 papers. Representative work includes from text to translation, using language models to prioritize variants for clinical review, Eigenlipids for exploring lipid biology and shaping the microvascular network, insights into skeletal muscle angiogenesis. Trend 3 of 10. Health services and behavioral research. It unifies research on health service delivery, behavioral interventions, and their impact on diverse populations and health outcomes. This cluster covers 431 papers. Representative work includes a scoping review and content analysis of available chronic pain education programs and resources for children, adolescents, and young adults with diverse abilities, the impact and application exercises on vocal fatigue, and economic evidence on provider-level implications of direct posterior amalgam alternatives following the EU phase-out. A scoping review. Trend 4 of 10. Microbiome and infectious disease. This trend explores the intricate roles of microbial communities and pathogens in infectious diseases and host interactions. This cluster covers 291 papers. Representative work includes gut-Hi-C metagenomes of severe COVID-19 patients, bacteria and yeast involved in gut-lung axis, the middle ear-nasopharyngeal microbiome axis associated with obstructive eustachian tube dysfunction in chronic otitis media, and molecular epidemiology and PFG-based genetic diversity of Clostridium perfringens in broiler chickens from Eastern Turkey. Trend 5 of 10. Cancer biology and therapeutics research. This trend investigates cancer's biology, mechanisms, diagnostics, and therapeutics to improve patient care and outcomes. This cluster covers 437 papers. Representative work includes futility in adenosquamous pancreatic cancer, radiomics-based prediction model for lymph node metastasis in thyroid microcarcinoma, and effects of luteolin on proliferation and apoptosis of breast cancer cells by regulating EGFR/STAT3/AKT pathway. Trend 6 of 10. Population health and health disparities. Exploring health outcomes and disparities across diverse populations, demographics, and clinical determinants. This cluster covers 300 papers. Representative work includes a systematic review and meta-analysis on the association between sarcopenia and cognitive impairment in middle-aged and older adults from Latin America, sex disparities in young adults aged 20 to 40 with acute coronary syndrome, and sex differences in cardiac structure and function following ST segment elevation myocardial infarction. Trend 7 of 10. Neuroscience and neurological disorders. Advances in understanding, diagnosing, and treating neurological and psychiatric conditions. This cluster covers 344 papers. Representative work includes artificial intelligence and neurodegeneration, state-of-the-art and explainability, instrumental assessment of sit-to-stand in Parkinson's disease, a scoping review, and porphyromonas gingivalis outer membrane vesicles drive neuroinflammation via TGM2, mediated mitochondria-associated endoplasmic reticulum membranes. Trend 8 of 10. Advanced computational and physical science. This trend integrates advanced computational methods with physical sciences for innovative analysis, prediction, and discovery across diverse domains. This cluster covers 558 papers. Representative work includes a tension-augmented hybrid framework with evolutionary optimization for robust deepfake detection, bridged scaffold editing of carbocycles and heterocycles, and a benchmark study of force fields implemented in CSD software. Trend 9 of 10. Clinical conditions and patient management. This trend investigates diverse clinical conditions, their diagnosis, epidemiology, and approaches to patient care management. This cluster covers 368 papers. Representative work includes serum and urine nerve growth factor and glycosaminoglycan levels in obstructive and non-obstructive feline urolithiasis and interstitial cystitis. Is there a socioeconomic and demographic correlation of non-accidental traumatic retinal hemorrhages and neurological complications after pediatric liver and kidney transplantation? A comprehensive review of acute symptomatic seizures. Trend 10 of 10, Public Health and Global Health. This trend analyzes diverse global and regional public health issues, their determinants, and impacts on specific populations and contexts. This cluster covers 280 papers. Representative work includes patient characteristics and healthcare resource use among patients with COVID-19 receiving treatment with nirmatrelvir/ritonavir, Paxlovid, in the Kingdom of Saudi Arabia, prevalence of antibiotic residues in red meat across Mogadishu, Somalia, and determinants of scabies among households in Fujita Likoma District, northwest Ethiopia. A mixed method study. Up next, our first paper out of 7, Safety, Tolerability, and Immunogenicity of Vaccine ALZ. 101 in patients with early Alzheimer's disease. Randomized controlled trial by Zofia Lovro and Juha Orine from Clinical Research Services, Turku, CRSD Oy, part of Link Medical, Turku, and Helsinki, Finland. Let's break down its key contributions. Alzheimer's disease is a progressive and debilitating condition with limited treatment options. This study investigates ALZ 101, a novel vaccine designed to target harmful amyloid beta oligomers, which are believed to play a key role in the disease. The core question was whether this vaccine is safe, tolerable, and can stimulate an immune response in patients with early Alzheimer's, potentially offering a new path forward. This was a Phase 1b randomized, double-blind, placebo-controlled trial involving 32 participants with early Alzheimer's disease. Participants received varying doses of the ALZ 101 vaccine or a placebo over several weeks. The main goals were to assess the vaccine's safety and how well it was tolerated, alongside measuring the immune response it generated. We also looked at exploratory biomarkers and cognitive changes to get an early sense of its potential impact. The study found that ALZ 101 was generally safe and well tolerated, with no serious adverse drug reactions reported. While some asymptomatic RAH events occurred, they were manageable. Crucially, the vaccine successfully induced robust, specific antibody responses against amyloid beta in almost all vaccinated individuals. Although no statistically significant differences in cognitive or biomarker outcomes were seen in the initial randomized phase, longer-term exploratory data hinted at potentially positive trends in CSF biomarkers and cognitive scores, though these were not statistically significant. In conclusion, this Phase 1 study provides encouraging evidence that ALZ 101 is safe, well-tolerated, and effectively stimulates an immune response against amyloid beta. While formal efficacy wasn't proven in this small trial, the exploratory biomarker trends are promising and warrant further investigation. These results strongly support advancing ALZ 101 into larger, adequately powered clinical trials to definitively assess its potential as a disease-modifying treatment for Alzheimer's disease. Up next, our second paper out of 7. Non-lytic viral immunotherapy induces long-term glioblastoma survival and tumor-specific immunity without eliciting an antiviral response by Alexander F. Haddad and Manish K. Aggie from Department of Neurological Surgery, University of California, San Francisco, California, USA. Let's break down its key contributions. Glioblastoma is a devastating brain cancer, known for its aggressive nature and resistance to conventional treatments. Patients face a grim prognosis, highlighting the critical need for innovative therapeutic approaches. This paper explores a novel strategy to tackle this challenge. Researchers developed a unique non-lytic retrovirus, RRVRLI, designed to deliver an immune-stimulating protein directly into glioblastoma cells. They tested this innovative therapy in orthotopic mouse models, aiming to turn tumors into immune-activating hubs and observe its impact on tumor growth and survival. The results were striking. RRVRLI dramatically suppressed tumor growth and significantly prolonged survival in the mouse models, even leading to lasting remission. We observed a strong immune response, with increased CD8 plus T cells and natural killer cells infiltrating the tumors. Importantly, combining RRVRLI with standard chemotherapy further enhanced this anti-tumor immunity, and the T cells were specifically targeting tumor cells, not the virus. This study presents a highly promising new viral immunotherapy for glioblastoma, effectively turning these aggressive tumors into immune-stimulating centers. While these findings are from mouse models, they pave the way for future human trials. This innovative strategy could potentially revolutionize treatment for glioblastoma and other challenging solid tumors. Up next, our third paper out of 7, Integrating Speech Biomarkers and Large Language Models for Adolescent Suicide Risk Detection with Mobile Application for Real-World Evaluation by Chong Lei and Runsin Shun from Vank School of Public Health, Tsinghua University, Beijing, China. Let's break down its key contributions. Adolescent suicide is a devastating public health crisis, and finding effective ways to identify at-risk youth is paramount. This research tackles that challenge by exploring a novel approach, using speech patterns combined with advanced AI to detect suicide risk. Researchers designed a two-phase study. They first developed their model using speech recordings from over 1,200 adolescents in structured settings. Then, they rigorously tested it on a separate group of 460 adolescents, collecting data naturally through a mobile app to see how it performed in real-world conditions. The results were very promising. The model achieved an accuracy of 0.808 and a macro F1 score of 0.807 for suicide risk detection. Importantly, it performed best on self-introduction tasks and maintained its effectiveness even when data was collected naturally through a mobile application. This study highlights a significant step forward. Integrating speech analysis with AI can provide a scalable tool for early suicide risk detection in adolescents. The success of the mobile app approach opens doors for widespread, naturalistic screening. Future work should focus on clinical implementation and assessing its real-world impact on saving lives. Up next, our fourth paper out of 7, Dual CRNK cells targeting PD-L1 and ERB2, HER2, exhibit cooperative cellular signaling and counteract solid tumor heterogeneity by Kristen Freudenberg-John and Yuri Eitler from Experimental Transfusion Medicine, Faculty of Medicine Carl Gustav Karras, Dresden University of Technology, Dresden, Germany. Let's break down its key contributions. Solid tumors are notoriously difficult to treat with current immunotherapies, including CRNK cells. This is largely due to the tumor's ability to hide from the immune system through varied antigen expression and by creating an environment that suppresses immune responses. This paper investigates whether a novel approach using dual-targeted CRNK cells can overcome these significant barriers. Researchers engineered NK92 cells to express two CRs, one targeting PD, L1, and another ERB2. They then rigorously tested these dual CRNK cells across a wide range of solid tumor models, including various cancer cell lines, 3D spheroids, patient-derived cells, and even in animal xenograft models. Their goal was to assess the cell's ability to kill cancer cells and understand the underlying mechanisms. The results were impressive. These dual CRNK cells showed potent and selective killing of various cancer cells, even when one of the target antigens was missing or blocked, which is a common problem in tumors. Mechanistically, they found that both CARs work together, boosting the signaling pathways inside the NK cells. Crucially, in animal models, these dual-targeted cells significantly outperformed single-target CRNK cells. This study provides compelling evidence that dual CRNK cells can effectively tackle the complexities of solid tumors, offering a robust strategy against antigen heterogeneity and immune evasion. While these findings are currently preclinical, they represent a significant step forward in developing more effective immunotherapies. The next crucial step will be to move this promising and adaptable platform towards clinical translation, bringing hope for patients with challenging solid tumors. Up next, our fifth paper out of 7, Enterococcus hyrae QD4713. Derived dopamine ameliorates intestinal inflammation and MPP-induced Parkinson's disease in mice by Tingting Zhao and Xiaodan Huang for National Engineering Laboratory for AIDS Vaccine, School of Life Sciences, Jilin University, Changchun, China. Let's break down its key contributions. Parkinson's disease, or PD, is a debilitating neurological disorder, and growing evidence points to a strong connection with our gut microbiome. Specifically, imbalances in gut bacteria can disrupt dopamine metabolism, a key factor in PD pathology. This highlights an urgent need to discover novel microbial interventions that can target this gut-brain axis. This study investigates a unique bacterial strain with potential therapeutic benefits. Researchers isolated a unique bacterial strain, Enterococcus hyrae QD4713, from the high-altitude Qinghai Tibet Plateau. They used a combination of advanced techniques, including whole genome sequencing, metabolomics, and various assays, to understand its effects. The study utilized both acute toxin-induced and genetic mouse models of Parkinson's disease to thoroughly evaluate the strain's therapeutic potential. This comprehensive approach allowed for a deep dive into both the mechanisms and the functional outcomes. The findings were quite compelling. Whole genome sequencing confirmed that QD4713 possesses a tyrosine decarboxylase gene, enabling it to produce dopamine. In mice, this led to a significant increase in L-DOPA and dopamine levels in both the colon and feces. Crucially, in Parkinson's disease mouse models, QD4713 not only improved motor performance, but also reduced dopaminergic neuron loss and increased dopamine levels in the brain striatum. These results strongly support its therapeutic potential. This research highlights the exciting potential of specific gut microbes, like Enterococcus hyrae QD4713, as a novel therapeutic strategy for Parkinson's disease. The discovery of its dopamine-producing capability offers a clear mechanistic pathway for its observed benefits. While these mouse model results are highly encouraging, the next critical step will be to translate these findings into human clinical trials to confirm safety and efficacy. This opens new avenues for microbiome-based interventions in neurodegenerative diseases. Up next, our sixth paper out of 7, Bionic Wearable ECG with Multimodal Large Language Models. Coherent Temporal Modeling for Early Ischemia Warning and Reperfusion Risk Stratification by Song Tao and Dong Deng from National Health Commission Key Laboratory of Cardiovascular Regenerative Medicine, Central China Subcenter of National Center for Cardiovascular Diseases, Hunan Cardiovascular Disease Center, Fuwai Central China Cardiovascular Hospital, Central China Fuwai Hospital of Zhengzhou, University, Zhengzhou 450046, PR China. Let's break down its key contributions. Myocardial ischemia is a critical health challenge globally, demanding better tools for early detection and risk assessment after treatment. While wearable ECGs show promise, they often fall short in understanding the intricate timing of heart events and predicting complications like reperfusion injury. This research aims to bridge that gap by developing a smarter, more capable system. Our team developed a novel framework combining bionic wearable ECG sensors with advanced multimodal large language models. This system uses a unique temporally hierarchical fusion transformer, designed with a cross-granularity attention mechanism. This allows it to simultaneously analyze intrabeat, interbeat, and long-term ECG dependencies, providing a comprehensive view of cardiac activity. We validated this innovative approach using 4 extensive datasets, encompassing over 100,000 patients. The results were truly impressive. Our model achieved an outstanding OROC of 0.947 for ischemia detection and a strong C-index of 0.923 for post-reperfusion risk stratification. This represents a significant improvement, with a relative OROC gain of 4.8% to 9.5% over existing baselines. Crucially, the system provided an average lead time of 18.4 minutes before an ischemic event, offering a vital window for clinical intervention. This research introduces a groundbreaking prototype for intelligent cardiovascular care, offering a new paradigm for early ischemia warning and reperfusion risk stratification. It successfully overcomes the limitations of current wearable technologies, which often struggle with the intricate temporal patterns of cardiac events. Looking ahead, the next steps involve rigorous prospective validation in real-world settings and seamless integration of this system into clinical decision support tools to truly revolutionize patient care. Up next, our 7th paper out of 7. Nanoplastics impair anti-tumor NK and CD8 T-cell cytotoxicity and antiviral B-cell antibody responses to SARS-CoV-2 by Stefano Maria Santini and Lucia Gabriele from Department of Oncology and Molecular Medicine, Istituto Superiore di Sanità, 00161, Rome, Italy. Let's break down its key contributions. Nanoplastics are everywhere in our environment, and we're constantly exposed to them. While we know they can cause health problems, their specific effects on our immune system, which protects us from infections and cancer, are not well understood. This knowledge gap is particularly important for patients undergoing immune-based therapies. Researchers use human immune cells, specifically PBMCs from healthy donors, as their model system. They expose these cells to oxidized and plasma-coated nanoplastics in a lab setting. The study employed various functional assays to assess how nanoplastics affect the activation and function of T cells, B cells, and natural killer cells, looking for changes in anti-tumor and antiviral responses, and exploring cellular mechanisms. The results showed that nanoplastics significantly impaired immune cell function. We observed reduced activation and functionality across T cells, B cells, and natural killer cells. Specifically, NK cells showed a decreased ability to kill tumor cells, and B cells produced fewer antibodies against SARS-CoV-2 after internalizing nanoplastics. These effects were also linked to increased immune cell interactions with the nanoplastics over time. This study highlights that nanoplastics pose a significant risk by weakening our immune system's ability to fight off infections and cancer. It also suggests that nanoplastic exposure could reduce the effectiveness of vital immune-based treatments. Moving forward, it's crucial to investigate these effects in living organisms and understand the long-term health implications of widespread nanoplastic contamination. That's all for today's PubMed Daily. If you found this helpful, please leave a comment, and subscribe for daily paper summaries. See you tomorrow.